3 weeks ago, a colleague of mine submitted a paper. Clinical diagnostics, patient samples. The whole thing hinged on a single PCR result that said the gene was present. Reviewers asked for a repeat. He ran it again. Gene was gone. Completely absent. Same DNA, same primers, same machine. Two opposite results from the same experiment.
The sample hadn't changed.
The sample had not changed. So what happened?
Did the machine break?
Machine was fine.
It is mildly likely the problem preceded the machine entirely.
It did! The problem was a single tube of enzyme that had been left on the bench for 40 minutes while he answered an email. That's it. That's the entire disaster. 40 minutes. Room temperature. Enzyme dead.
And how many reagent kits in that paper? Because a retraction doesn't just cost time.
The point is, PCR looks simple. 2 primers, some enzyme, a thermocycler, done. But the reason it fails, when it fails, is almost always something invisible. Something that happened before you hit start. Today we're going to understand this technique well enough that you never get caught out by an email again.
PCR, polymerase chain. Reactioneer is a method for amplifying a specific sequence of DNA exponentially from a tiny starting amount. In theory, a single molecule of template DNA is enough. The principle is enzymatic copying. A heat-stable DNA polymerase extends a pair of short, synthetic primers that flank your sequence of interest, producing a double-stranded copy. That copy becomes a template. Each cycle doubles the product. After 30 cycles, you have over 1 billion copies of a sequence that originally existed in nanogram quantities.
Molecular photocopier. That's the analogy. You're not inventing DNA. You're amplifying what's already there.
And like a photocopier, what you put in determines what comes out. Garbage template produces garbage amplicon, just in vast quantities.
And if you copy the wrong thing in vast quantities, you've wasted a kit that costs 40 euros a run. At scale, that mounts fast.
If you want to keep doing this every Monday, subscribe. The bell icon. Actually use it. I have protocols to fund and a thermocycler that isn't going to pay for itself. Equipment. Here's what you need and here's what you can't cheap out on. Thermocycler. Any modern block cycler works, but calibration matters. If your block has a 2-degree temperature gradient across it, your annealing is inconsistent across lanes, and you'll get differential results from the same plate. Check the calibration records. TAC or high-fidelity polymerase. TAC is cheap and robust. High-fidelity, Fusion, Q5, whatever your lab uses, is slower but has proofreading. If you're cloning the product, you use high-fidelity. If you're just checking whether a band is present, TAC is fine. Do not mix them up.
I use the TAC from the -20 that's been defrosting since Tuesday.
No, stop. When did it defrost?
It was on the bench when I arrived.
That enzyme is dead. TAC loses activity fast at room temperature. Anything above 4 degrees for more than a few hours and you're gambling. You're not gambling today.
TAC polymerase is a thermostable enzyme, but thermostable does not mean thermally indestructible at ambient temperature over extended time. Prolonged exposure to room temperature causes irreversible denaturation of the active site. The enzyme may still enter solution and appear functional, but KCAT drops dramatically. You will see either no band or a faint smear, both of which waste your template.
It is mildly ironic that the enzyme designed to survive 95 degrees cannot survive a Tuesday morning.
Primers. Desalted is fine for most applications. HPLC purified if you're doing sensitive detection work. Keep them as 10 micromolar working stocks. Do not repeatedly freeze-thaw your primer stocks. Make small aliquots. Label them clearly, not F and R. Write the actual name. DNTPs pre-mixed is convenient, but check the concentration. Most master mixes have them included. If you're building your own reaction, you want 200 micromolar each in the final volume. More is not better. Excess DNTPs chelate magnesium, and you'll tank your polymerase activity.
Pre-mixed master mixes cost more upfront, but they're cheaper than troubleshooting a homemade reaction for 3 days.
Template DNA. This is the one people underestimate. Quality matters more than quantity. You want clean DNA, good A260-280 ratio, no phenol carryover, no ethanol residue from your extraction. Quantity for a standard reaction is 1 to 50 nanograms of genomic DNA, or 1 to 10 nanograms of plasmid. Don't eyeball it. Quantify it. Magnesium. If you're using a commercial master mix, magnesium is already in there at optimal concentration. If you're building from scratch, start at 1.5 millimolar and optimize up or down if you're getting no band or non-specific bands. Magnesium is the cofactor for the polymerase. Get it wrong and nothing works regardless of how good your primers are. Protocol. Step 1. Reaction setup. Do this on ice. Always on ice. You are preventing any premature extension before your first denaturation cycle. Master mix first, template last. The reason is simple. Master mix is the same across all your reactions. Template is the variable. Add it last. Close the tube, and you know exactly what went into each reaction.
The reason ice matters mechanistically. TAC has some low-level activity even at room temperature. If your primers find even a partially complementary region and begin extending before the first denaturation step, you generate nonspecific products that carry through all 30 cycles. Hot start polymerases exist specifically to block this. They use an antibody or chemical modification to inhibit the enzyme below 60 degrees. But even with hot start, keep it cold. There is no upside to warmth during setup.
I set up on the bench. It's fine. It's only been 10 minutes.
10 minutes is long enough, especially with TAC, especially if your primers have any secondary structure. Ice every time.
Additionally, 10 minutes at room temperature with open tubes in a PCR-heavy lab is a contamination window. PCR products from previous runs are airborne at low levels. Your negative control will tell you whether you've picked something up, which is why the negative control is non-negotiable.
It is mildly unsettling how many PCR setups occur without a negative control.
Step 2. Thermocycler program. Initial denaturation. 95 degrees for 2 to 3 minutes. This is not the same as your cycling denaturation step. This is specifically to fully linearize your template and if you're using a hot start enzyme to activate it. Do not skip it and do not shorten it for genomic DNA templates.
Genomic DNA is packaged with histones and higher order chromatin structure. Even after extraction, it retains some compaction. The initial denaturation step disrupts that residual structure, making the template fully accessible to primers. A 30-second initial denaturation that works for plasmid DNA will give you weak or absent product from genomic template.
Cycling denaturation, 95 degrees for 30 seconds. Melts your double-stranded DNA into single strands, every cycle. This is where your enzyme has to survive, which is why you need thermostable polymerase and not the stuff from your kitchen. Annealing. Here is where people get it wrong the most. The annealing temperature should be approximately 5 degrees below the TM of your primers. Your primer design software will give you a TM. Trust it as a starting point. Do not trust the protocol sheet that came with your kit. That's a generic number. Your primers have a specific TM. Use it.
Annealing works because at temperatures slightly below the TM, the equilibrium favors primer-template hybridization. The polymerase can only extend from a stably annealed, double-stranded primer-template junction. It cannot initiate synthesis on single-stranded DNA alone. Too high an annealing temperature and the primers won't bind. Too low and they bind everywhere non-specifically, giving you a gel that looks like a smear.
Gradient PCR exists because nobody wants to run 12 optimization reactions at 40 euros a plate. One gradient block, one run, you've got your temperature.
Extension, 72 degrees. This is the optimal temperature for TAC. Extension time depends on your product size. Rule of thumb, 1 minute per kilobase for TAC, 30 seconds per kilobase for high-fidelity enzymes. Do not extend longer than you need to. You accumulate errors, and you give your polymerase time to add non-specific bases to the ends of your product. Final extension, 72 degrees for 5 to 10 minutes after your last cycle. This completes any partially extended strands. Skip it and your product will have frayed ends. That matters if you're cloning. Cycle number, 30 to 35 cycles for most applications. More than 40, and you're amplifying contamination. If you need more than 40 cycles to see a product, your template quality is the problem, not the cycle count.
I set it to 45 cycles, just to be sure.
45 cycles means you're amplifying noise. If your band is real, you'll see it at 32. If you need 45, the band is not real. It's a contaminant or a primer-dimer. You've just made a lot of something you don't want.
Exponential amplification means that at 45 cycles, you're generating over 30 trillion copies from a single starting molecule. At that scale, any spurious priming event, a primer-dimer, a mispriming on genomic background, becomes visible on a gel. The signal-to-noise ratio collapses. Reduce your cycles and fix your template, not the cycle count.
It is mildly characteristic of PCR that the solution to seeing nothing is never more cycles.
Step 3. Run your gel. After PCR, you load your product on an agarose gel alongside a DNA ladder. You're looking for a single band at the expected size. Clean, sharp. One band.
What good data looks like. A single, bright band at the correct molecular weight. Your positive control should show the same. Your negative control, the reaction with water instead of template, should show nothing. No band in the negative control means your reagents are clean and your setup was aseptic. A band in the negative control means contamination, and your positive result cannot be trusted.
What bad data looks like. Smear instead of a band means non-specific amplification, usually annealing temperature too low or too many cycles. Multiple bands mean the same, plus possibly mispriming. No band means enzyme failure, template quality issue, or primer design problem. A band in your negative control means you stop everything and decontaminate before running anything again.
A contaminated negative control in a diagnostic run is not a troubleshooting note. It's a retraction risk. The cost of that is not reagents.
Applications. PCR is the technique that made modern molecular biology possible. Diagnostics. COVID tests, HIV viral load, cancer mutation screening, research, checking transgene integration, genotyping mouse colonies, verifying plasmid inserts. Forensics, paternity testing, ancient DNA from a 40,000-year-old bone. Every one of those applications runs on the same 3-step cycle we just walked through.
The reason PCR is foundational is specificity and sensitivity in combination. 2 primers, each 20 bases, define a unique sequence in a genome of 3 billion base pairs. The probability of a random match is astronomically low. Combined with exponential amplification, you can detect a single copy of a sequence in a background of billions of unrelated molecules. No other technique achieves that combination without PCR as a first step.
Now, about the thermocycler in my shed.
Quel but?
It's a legitimate instrument. I bought it from a university surplus sale. It's calibrated. I calibrated it myself with a thermocouple and a spreadsheet.
I want to be clear that I'm not aware of this.
I've been running a PCR-based genotyping panel for the last 6 weeks. On samples I won't specify from a source I also won't specify.
Cycle 92 completed this morning. I changed the buffer.
She's been running the overnight cycles. We're at a scale that I would describe as ambitious.
It is mildly alarming that the calibration instrument is a spreadsheet.
Closing tip. When your PCR fails, start with the negative control and work backwards. If your negative has a band, stop its contamination and nothing else matters. If your negative is clean and your positive has no band, check your enzyme first, then your template, then your primers, in that order. The thermocycler is almost never the problem. The problem is almost always in the tube before you closed it. Method Monday, every Monday.